cross-section of the stomach. Because what you can find in one piece, that is exactly the same, except that you can find the cavity or the space for the food to pass through. Who is your lecturer? Alright, so that is why we represent one system. You understand? So this is one sector of the entire cross section. Okay, now because of the microscopic examination, then we are going to identify the parts of the structure. Right? Now, starting from the outermost layer, then you will have the tonica serosa. Did you place it when you come in the drawing, or you only place it? T could stand for Ella, T for stand for tonica. So you have to take note of what is T. And that is why we know now this tonica serosa is actually a continuation of the visceral peritoneum. You will have your weakness. The visceral peritoneum covers all the outer part of the internal organ. Okay. Now, so you have the tonica serosa. Now, going inward, then you will find the tonica follicularis, which is composed of 2 layers. The outer is longitudinal, and the inner is what? The inner is circular. Now you might think, why when it is round and it is longitudinal? Well, it is because if you have a longitudinal muscle, okay. Now remember, this is a cross-section. So if you cut that cross-sectionally, so what will be the cross-section? You will have round, correct? Now, if you have a round structure, and you cut this cross-sectionally, so it will appear yes. So do not think of the shape, but you have to understand the structure. Now, do you understand? So this is the longitudinal because this is cross-section, and this is the circular because of this cross-section. So you do not have to identify based on the shape, but on the structured parts. So in the practical exam, if this is focused, that is not circular, correct? And if this is focused, that is not longitudinal, because our specimen is a cross section. So that is why you have to pay attention to our pre-lab. So do you know that why it appears this one and the round? Before we explain, you will say that this is the circular because of the shape, but that is not so. That is why it is important that you read and understand what you write in your laboratory. So if you identify this as circular, that's not correct. On Friday, and this is not longitudinal, but that is circular. Then you might say, oh, how come that the shape does not indicate? Because this is a structure. It's not shapes and sizes that you do in kindergarten. Alright, so going towards the inside, then you will find that there is an elevation, and the elevation is known as the root. Right? Now, then beyond the tonica muscularis, you will find loose connective tissue. And at the same time, you will find the cross-section of a blood vessel. And you have the cross-section of a lymph vessel. What is the difference between the two? You have red blood cells, right? Or blood cells, but this one is an empty space. Are you following? Now we go to the inner part. So it is not drawing, but you read your laboratory manual. Because you are late in your lecture, correct? This should have been more or less this together in the lecture. You are still in the muscular system. And that's still a way off. And we only have 3 minutes before the final. No, we have had so many vacations. Alright. So you have, this is the tela submucosa. Where you find the lymph, the lactic tissue, and the blood. Okay. Now beyond this, then you have the tunica mucosa. Now the tunica mucosa is composed of the following parts. Number 1 is the epithelium. Now these are the columnar cells that make up the ruga. Correct? All of these are columnar. Okay? Now then the spaces between the columnar cells, these are the gastric pit, meaning the opening towards the gastric gland. Are you following? So you have here the gastric pit, you have another one, not pit, I said pit. Oh, P-I-T. Okay, so that is one. The epithelium is composed of columnar cells. So that is one part of the tunica mucosa. Now the second part, say you have this red. And you find it at the base of the columnar cells. And this is the lamina propria that is found in your mouth. Okay? Now beyond this lamina propria, what are these round things? These are the longitudinal muscles. And what are these dashed lines? These are the circular. So aside from the muscle layer, we also have another muscle layer in the tunica. But we do not call it as tunica muscularis, but we call it as muscularis mucosa. Now, only the stomach. Now, will you please pay attention? Now, please pay attention. You know, in kindergarten, if somebody is talking, or I will stop and will tell all about it. We will have only 2 more practical exams. Okay? But anyway, kindly pay attention because we are going to finish all this afternoon. And you need an orientation to understand the microscope. Alright. So you have the outer is, what is this?
Circular.
Then this one?
Longitudinal.
Both are known as mucosa. Here it is tunica mucosa. Are you following? So you draw one ruga. I repeat, what you find in one ruga is true with all the other parts. And I gave you an analogy. When we eat the cake, you don't eat the whole cake, but we only get a slice. Because what you find here and what you tasted is true of all the other parts. Of course, we do not eat stomach. No, we have, yes, it is fun. There is there for that. We eat the stomach, the intestine, what is that? The one that are selling near the center. Oh. Okay, now, do you have any questions so far? Now, let's take up the small intestine. Okay. Now, if you have the large stomach, okay, then this is followed by the small diameter of the intestine. Correct? So, if it is smaller, then the cross-section would be like this. Correct? Oh. This is cross-section of the stomach because it is wide. And you have smaller cross-section because the intestine has a narrow diameter. Okay. So you cut this again cross-sectionally. And you will find this finger-like elevations. Now, the finger-like elevations of the small intestine are known as the villi. Okay. Singular term, you have villus. So one ruga, so many rugae. One vilos, many villi. So that's the plural term. Okay? So again, we take up only one vilos to identify the parts. As I said, you don't have to eat the whole cake. But what you find in one sector will be true with all the other parts. Okay, so we have the cross-section of the small intestine. Okay. Now, just like the stomach, because the contraction of the tunica muscularis will enable the food to push down to the lower part of the digestive system, correct? Now, the outermost is the tunica serosa, followed by the longitudinal layer. Then you have
Circular layer.
the circular layer. And both are known as
Tunica
muscularis.
muscularis.
Okay. Now then you have the tela submucosa and the tunica mucosa. There is no muscularis mucosa in the small intestine. You have only one layer of columnar cells. That is the nucleus, okay? Yes. So only one layer, unlike in the stomach, wherein the ruga has so many columnar cells. Okay. Now, among the ordinary columnar cells, then you will find scattered, okay? Scattered among the ordinary columnar cells, then you find a cup-shaped depression. Now, if you find a columnar cell with this cup-like depression, this is a goblet cell. Now, a goblet cell is a unicellular gland. Okay? Goblet cell. So, this is the cross-section of the small intestine. This is focused under the microscope. We do not care about your drawings. What we do care is for you to understand the parts. Because this is what you will see again in your practical exam. Okay, then we go to the liver. You take only a part of the liver. Now, liver cells are known as hepatocyte. Okay? Now, then you will find the blood capillary. You always can identify blood capillary or blood vessel because of the presence of the blood cells. Okay. Now, near this, then you have the bile capillary. You have a space in the center, but no blood cells. And they are much smaller. This one is elongated. Okay. Now, scattered among the hepatocytes, then you find dark brown pigments. And these are the pigment cells or pigment cells, which are responsible for the color of bile. You know what is bile? So this is the digestive system. Now, then we go to the urogenita, which you again, it's all focused. You kindly turn the blackboard. Tiny tubes. So, if it is a tube, so the cross-section is always round, correct? With a space in the center. However, because it is coiled, we have millions, including these tubules in our kidney. So, coiled mana. So, ana, this one, this, okay. Now, if the section falls under this portion, so it becomes round, correct? Now, what about if it occurs along the curvature? So, it will be something like oval, right? Now, however, this is a diagrammatic representation. Now, all of this represents the millions of very tiny tubes in our kidneys. Our kidneys will extract the soluble waste product from the blood. Now, if our kidneys fail to remove the waste products, then we have what? We undergo
Dialysis.
or kidney transplant because that will be poisonous to our blood, and so we end up in some memorial garden. Diba?
I see.
So we have this section. Now all of this are the millions of what? Of uriniferous tubules. Now, in relation to the tubules, then you have the functional unit. Our blood enters into the kidney, then into the blood vessel by the process of diffusion, filtration, then the waste products of the blood will pass along this through the mesonephric duct to the urinary bladder to the outside. That is why we extract urine. Diba? So if our kidneys cannot function, how can we get rid of this waste products carried by the blood? So we die of what? Then one of my students, we only die. Okay? So you have the kidney. Now, scattered, these are the functional parts of the kidney. And these are what we call as the Bowman's capsule, this surrounding part, okay? Then you have the blood cells, okay? And this is what we call as the glomerulus. Okay, now these are the blood vessels that are enclosed with the Bowman's capsule. The Bowman's capsule plus glomerulus, then that's the functional part of the kidney. And we call it as the Malpighian body. Malpighian body. Or we call it as the renal corpuscle. So, a Malpighian body or a renal corpuscle is composed of glomerulus and the Bowman's capsule. Just to give you an idea of what they are. Okay? So under the microscope, if we focus it in the center, that is the glomerulus. If it is outside that shape, that is the Bowman's capsule. Both are parts of the Malpighian body or the renal corpuscle. Okay? Alright. Now, alongside the kidney, you have the mesonephric duct. Now, if it is mesonephric, it's actually a duct, so it's a passageway. So it is what? A round structure, and this is the cavity for the passage of urine. Okay. Now, beside, you have the renal portal vein. Because it is a vein, so you find blood cells. Okay. So you have mesonephric, they always are found side by side. Because if you have the kidney, that is the renal portal. Then you have the mesonephric duct that are side by side. So if you cut this cross-sectionally, so you have side by side. Okay? So all of these are also focused. Right? Now then we go to the ovary, the female. Now you can identify all of the cells because of the size and the allocation of the numbers. You have oocnia, small cells, nucleus, etc. Then you have the young oocyte, bigger cells, and of course also a big nucleus in the center. Now in the case of the old oocyte, much bigger cells, but the nucleus is not found in the center. Okay, and you have yolk material, or we call it as yolk materials. Okay, so this will be taken up after digestion. Now let us see what is the testis. Alright, you know the testis is found where? Attached to the kidney, right? Then you cut this cross-sectionally again. And And this is what you will find. Okay? There are compartments. One compartment will have this. And all of this will present the development of the sperm cell or spermatozoa. Spermatozoa is plural. Okay? Spermatozoon. Now, you will find, like for example, one cell. Okay? And they're found near the outer part of this space. And the nucleus is in the center. And they always form groups. There. Now, the bigger cells, this, and it has granular nucleus. This is the spermatogonium. This is the spermatocyte. Okay. Now, then you find much smaller cells. And these are the spermatids. And hair-like structures in the center, these are the spermatozoa. Okay? Now, they have already a tail or a flagellum because the sperm cells are capable of movement. Whereas female germ cells cannot effect movement. So this is what you are going to identify. You only take note of the size of the cells. Okay? And these are all focused under the microscope. Okay, so you start with the digestive, or so that you will not group all at the same time. Then some of you work on the urogenital and the others on the digestive. So you can have this.